So a lot of the times it's healthy stuff about presentation, but sometimes it's can dig a little bit deeper just in terms of like different ways to present the data, different tones. So for the most part, I've always found it useful because, you know, we don't have to worry about no additional work. It's usually about putting in additional caveats to the data. Or like I said,
policy, right? I mean, they do advise reviewers against.
Yeah, and with ours, we're guaranteed the publication as long as we follow through. But it tends to be more on the not so much like, I don't want to use the word political, but it is kind of that side of it, right? Which is we're still communicating as individuals, and so just the tone, the language, what gets discussed, what doesn't get discussed. From my standpoint, you know, we start with all of these very, very systematically, and so they come out more technical, and then peer review essentially turns them into what they are. So all of the initial submissions, I do exactly the same. They're pretty bland and dry, and peer review, I let peer review shape it essentially. And I find that actually quite fascinating, how that happens, right? Because if you get
it's a lot of work, right? I mean, that's maybe something against putting out like everyone in different manuscripts. Like you have to go over the process.
Mm-hmm. It is a lot more work. It is a lot more work. It's just like anything else. Writing a paper is a lot more work than if we just skip that stuff.
That's for the interactive paper thing. I like that.
Yeah. That was something that happened in the background. Yeah, that was nice. That was a fun one to pick. We had to figure out which paper we were going to choose for that, because we do the same thing for every single paper. I'll talk about that briefly in the webinar. I think that's a fun thing about these projects is you can not just investigate what's happening, but you can also try to demonstrate practices at the same time. So
yeah,
I hope that has a lingering effect.
I think the same holds for us. So how's SCORE going? I mean, have you?
That's what's been consuming me. It's intense. It makes all of these other projects that we do look like nothing. It's so massive.
What's your starting point, though? I mean, like just like selecting a whole bunch of papers and predicting before, and then trying to select.
Yeah. So for that one, again, it's social behavioral broad spectrum, so that's fun. And we're going back to papers that are at least 10 years old, so a pretty large window. And we've got about 60 papers that we've identified. Oops, somebody's chatting. So the idea there is because it goes tiered down effect, because we can't replicate everything because it'd be way too costly even for DARPA. So we're just using random sampling to go from the journals that we selected and the years that we selected, and just kind of randomly selecting a subset of empirical articles that fit the criteria, and we just kind of funnel that down. Because what will happen is we can have more that we can do machine learning approaches on. By we, I mean DARPA, right? That's accessible to that. Less of that, but still quite a few that can be applied for humans doing prediction markets or export forecasting, and then less for replication because it's the most labor intensive. So we essentially just dial down 10 every single time, right? 30,000, 3,300. And we just do stratified random sampling through the journals in the years to try to maintain at least a good sampling strategy.
Well, your goal is 300 papers? That's a lot.
A lot, yeah. Yeah, they all vary though, because they're going to be different disciplines and different areas, right? Because like I said, we go from everything from like macroeconomics down into social psychology. So there's still some fine tuning in terms of, because there's strategy in terms of the way we order our journals. Because like we said, we just kind of using a stratified approach. The beauty of that is we can adjust if need be. So right now we're still just getting a little more clarity from DARPA on what they want. Because obviously we need to conform to the overall program's goals. But yeah, it's going to start. We've got a couple more little fine details to start out, and then we're going to start moving, which is, yeah, it's really fast.
Selecting experiments was actually harder than we thought to get a random sample, because I mean a lot of things get cut out at various points. In the end, we have expertise, right? I mean, even though we have 60 labs, it's still, I mean, it's not that easy to actually find an experiment that fits inclusion criteria and that we have like 4 or 5 labs that can replicate. So I mean, I think we needed to screen like 30. We did our first screening of like 15,000 papers, and some methods got like 15 of them included. So I mean, we had to download another 15,000. So it's been more work than I thought. I think we should be done by next week, but I mean, it's yeah, you lose stuff by all kinds of reasons.
Cool. Yeah.
Some experiments are complicated, right? I mean, it can be hard to. But do you have any idea of who's replicating, or just like getting like general thing over this looks vaguely?
So yeah. So for
and then go off.
No, yeah. For SCORE, it's going to be similar to kind of how you've got your approach, but not quite as upfront in terms of. We're going to narrow it down by the techniques. We're going to let that we're going to have the reverse happen. If we can't do something, it's because of the technique. So we're casting a wide net. But the same approach of let's get a large network of people that are interested in doing this, that are aligned. You know, we've done this before, and there's groups that are forming, and let's leverage them to identify, you know, how big of a net can we get, and you know, if it turns out that we lack expertise for a given technique or a given area, then so be it. Then we just don't do anything, right? Because we're not going to push the envelope. But otherwise, we're just casting a huge net. So we've got, you know.
And you do have a network, right? I mean, you can just go back to the people in RP.
Yeah. So we'll see.
That's technically constrained than ours in terms of like psychologists' experiments are generally more.
Yeah, a lot of it will hang. Some of it will hang on some of the experiments, like the cognitive experiments, some of it might get a little tricky, but you're right in the sense that most of it hangs out, not all of it, but a lot of it will hang out on analytical techniques. So it's more about, do we have somebody who can understand and do the analysis, which is pretty broad. And that's a big piece of the puzzle that we're trying to do. So hopefully it's not too, I'm sure we'll hit a couple of cases where we can't, maybe the participant recruitment's just really tricky in some areas. You can't get enough people. But yeah, I think you're right that I think we'll be able to cast a pretty wide net and be able to do quite a bit. Like I said, we're lucky in the sense that this has been kind of forming for a while. So we're just kind of amplifying networks that already exist, as opposed to what you had to do, which is create a brand new network, right? Which is a lot harder.
Maybe, I mean, that's what we'll be able to do in like 2025 or something.
Yeah, that's right. That's right. Yeah.
Or hopefully somebody else will do, right? I mean,
yeah.
I don't want to coordinate another one of this, but well, we're 3 minutes off from starting time. So I think we just wait for people to show up.
We are on live on Facebook for 10 minutes.
Oh, nice. Okay.
Okay. Hi, Tim.
Anything simple about the project, right? So yeah, we'll just wait for the right time. I think we're
yes.
And if people want to come on camera and chat with us, it's okay. But otherwise, when we start, we'll just let Tim online, and we can have everybody to just like ask questions and appear on camera afterwards if they want. If not, they can just throw the questions on by chat, and we'll send it to you. So yeah, let's just wait for 2 minutes. Yeah, we could just have the advertise our next lecture, but I think I can do that at the end, just like put on my screen. How many people do you have? Can we say 23? Okay, that's good. I think more people join. I mean, Brazilians are not like super timely as the stereotype goes, so we might have people joining in over the first 5 to 10 minutes. But we're 60. I think we're 60 something labs at the moment. But there's going to be people from outside as well. There's this one science reporter at the moment, so he's probably doing some news piece on us or something. But I recognize his name. Cool. But yeah, I think we can pretty much kick off. It's 3:59. So who's there from the initiative? I mean, can people like raise their hands or?
Yes, raise your hand. Yeah.
Or say they're around.
I think there is more. Yeah.
Oh, I see. Nice.
Okay. Okay, so when the clock is 4, I'll officially start. I'll do a short address in Portuguese before moving to you, just to chat with the people here. But we should wait for the official time. Yeah, so there is. Bom, gente, não sei quem está online aqui. Bem-vindos ao nosso primeiro webinário da Iniciativa Brasileira de Reprodutibilidade. Bem-vindos, enfim, pessoal do projeto. Acho que muitos dos presentes aqui são prestadores envolvidos na iniciativa. É o nosso público primário, afinal. Dito isso, a gente está aberto online para quem se inscreveu no webinário. A gente está aberto via Facebook. Vocês podem divulgar o link, inclusive, se vocês quiserem postar nas redes de vocês. Nossa ideia é para maximizar o número de pessoas que possam estar participando aí e tal. O vídeo deve ficar disponível depois no nosso canal no YouTube e no Facebook também. E, de novo, a ideia é ter esses webinários mensalmente para discutir temas relacionados à reprodutibilidade científica, que obviamente é um tema amplo, então pode ser visto de diferentes perspectivas, e vai ser muito legal começar hoje com o Tim Errington, que é do Center for Open Science dos Estados Unidos, que, bem ou mal, enfim, é o coordenador do Reproducibility Project, Cancer Biology, que foi talvez a inspiração mais próxima, ou a coisa mais perto do que a gente gostaria de fazer. E certamente acho que tem muito. Foi a primeira pessoa que a gente contatou quando a gente começou o projeto aí e tal, para pedir conselhos. Então acho que ele vai ter bastante a oferecer para a gente, assim, e tal, tanto no sentido do que deu certo, do que não deu. E acho que ouvir o tema é sempre estimulante e um pouco assustador, assim, porque se você se dá conta do quão desafiador a coisa pode ficar. So I'll switch to English now. I kind of presented you already, but I'd like to welcome Tim Errington from the Center for Open Science in the US, in Charlottesville, Virginia. He's this research director there. He's been the mastermind behind the Reproducibility Project Cancer Biology, which has been a large effort that is actually still ongoing because they're publishing results, but to replicate high-impact papers in the area of cancer biology. So he'll tell us a little bit about that, and I mean, what worked, what didn't, and what's still to come. He's also involved in SCORE, which is a big project to replicate, I mean, the huge replication, prediction/replication project on the social sciences that is starting right now with financing from DARPA in the US. And Will, I mean, we're very, very glad to have you here. Thanks a lot for accepting the invitation. So as we said, maybe our idea would have Tim to speak for like 15 or 20 minutes, then open up to questions because I think it makes things more interactive, and then let him have another 15 to 20 minutes, and we'll do questions again at the end. So thanks for being here, and I mean, it's all yours now. I'll turn off my mic and you can share your screen or whatever you want.
All right, wonderful. Thanks for the intro, and yeah, let me see if I can get this sharing real quick. Correct me if it's wrong. You should be able to see my presentation slides. I'm going to see if I can present them. Can you see that okay? Yeah?
Okay, Tim. It's okay.
Yep, it's great. Okay, great. All right, well, thanks for the intro. So I'm going to give you guys a rundown on a bit of what we're doing at the Center for Open Science and a focus on some of the work we're doing with the Reproducibility Project in cancer biology. So disclosing first the funders who've supported us over the years, particularly the Arnold Foundation, or Arnold Ventures, who's been the backer behind the cancer biology project that I'll talk about. So I thought what I'd start with is a little bit of a background on COS in general first, before we get into the project and why we started it and where we're at. As you heard, we're based in Charlottesville, Virginia, in the United States, and we have a broad mission to improve openness, integrity, and reproducibility of scientific research, and that's agnostic to discipline. So, you know, biology, you know, social sciences, basically interested in the research enterprise in itself. We do this in a couple activities. We do research, which is what I direct here at the center, or meta-science research, right? Research on research. You know, basically looking for evidence of what our current practices are and ways to evaluate new initiatives to encourage change. We also do a lot of policy work, largely with journals and funders, to kind of shift those incentives. And the backbone of a lot of what we work on is infrastructure. So, you know, developing free open source software to enable change to occur within researchers. So I think to give you a good idea of the work that we're doing is to kind of actually use an example of some values that were described by Robert Merton back in 1942. So I'll kind of go through this quickly, but this gives a good background of why we exist. So Robert Merton in 42 described the normative values of science, which you see on the left side of the screen. about open sharing, evaluating research on its merit, being motivated by knowledge and discovery, and being skeptical of all evidence, including your own. And counter to that are these counter norms that you described, which is, you can think of science as being secretive, evaluating it by reputation, what journal something's published in or what institution somebody's from, treating it as a competition and only advancing one's own theories and findings versus trying to kind of root through all the evidence. And even though Robert Merton didn't describe it this way, we can think of it a little bit as quality versus quantity. All right, so this is, just essentially the normative values, what scientists believe, and this is what, like I said, described in 1942. So in 2007, Anderson and colleagues decided to ask NIH-funded researchers what they subscribed to or what they believed in. Did they believe in those normative values or those counter norms, or both? And what we see here in this graph, in that solid gray that takes up 85+% of both mid-career and early career researchers, is that they subscribe to those normative values. They think those are more important to what they believe in, what they subscribe. The dark black there, way on the right, the small fraction, those are saying, no, I think those counter norms are more important than the normative values. And the hash stripe there, that's about 10%, is saying, no, I think they both are important in the way that I think about science. So that's not very exciting, right? That basically tells us, well, what Robert Merton described in 42 still existed in the population that Anderson and colleagues just, were investigating in 2007. So then Anderson and the colleagues, they said, well, don't tell me what you believe in, tell me what you practice. When you're actually conducting your research, what's more important, those normative values or those counter norms? And what you see is that it's largely the same, that we see alignment between what we believe and what we actually conduct, but that there is this growing recognition of, yeah, those counter norms actually do play a role in the way that I actually conduct my research. So then they said, well, don't tell me what you do in your own behavior. Tell me what everyone around you does. Tell me what your colleagues are doing, that are down the hall from you at your institution or in your subfield. And what you see is a complete reversal, right? You see that when we look at others' behaviors, we don't believe that they're, essentially playing by the same rules that we are. And what we see here is essentially what I think we're trying to do at the Center for Open Science, which is find an alignment between, the values that we practice and the incentives that kind of drive our practices. So I'm just going to go through a couple little more barriers, then kind of get right into the reason we started the Reproducibility Project in cancer biology. So, there's some barriers to the way that we conduct our research and why we need to investigate how we conduct our research, such as those perceived norms that I talked about. There's also motivated reasoning, right? That I'm basically here to promote basically my own biases. I got to advance my career. So if I, maybe write or present my results in a certain manner, it's not because I'm trying to be dishonest or I'm trying to confuse somebody. It's that I'm just trying to kind of get ahead on this. I do this in minimal accountability, right? Right now, when I publish a paper, unless I give you more, that's all you see is what's in my paper. And that's actually a really important part that I'm going to come back to in the Cancer Biology Project. Those concrete rewards, the paper, the grant, those beat these abstract principles of openness. Openness takes more time. Being kind of closed and just kind of hitting those concrete rewards is sometimes a bit easier. And that basically gets at a big barrier, right? Which is we're busy. And again, this is something else I'm going to come back to again when I talk about the Cancer Biology Project. It's just this kind of busy element that goes into the barriers that exist. And there's a big one. There's one that's hidden behind all this, especially in academia, which is those incentives for success tend to get more focused on getting it published, not necessarily kind of focusing on getting it right. All right, so we're not going to quite go in linear fashion here in terms of order, but there has been a lot of news about a quote unquote crisis. I'm going to use that word not because I agree that there's a crisis, more that that's, I think, what we're all talking about, or at least what's get covered in news articles and the such. And a good example of this, a well-done one, is by Baker in Nature, where they conducted an informal survey of a little over 1,500 researchers and just asked that open-ended question: is there a crisis? And what you found is that many people said yes, either because there's a significant one or a slight crisis, whatever that means, doesn't matter. But I think there is a growing recognition that there's something going on, or at least there's something that we're acknowledging that maybe we should be, being more attentive to. And it's a bit of what these projects that we and others have been conducting are essentially exposing. What's more interesting is I think really when we ask the question about have you failed to reproduce an experiment, and that one, it occurs across discipline. This is not unique to biology or psychology. It's unique to the sciences. And that it's not even unique to, I can't replicate somebody else's work, or I can't replicate my own work, right? That it applies to both at a pretty high rate. And I think that's actually quite interesting in itself. And it's something that we can kind of explore a bit more. And I'd love to get questions on this because it's something really hard to think about when we, what this actually means. So if we go back a little bit more farther behind before that survey, and we get back to 2009, 2010, 11, 12, there's a lot of news back then about there being a cause of concern in terms of at least the credibility of research, right? That we think research, that when we publish it, is this essentially bearer of truth. And we're finding out more and more is that it's not, right? That just because something's been published, just because it's gone through peer review, does not mean it's quote unquote true. Not to say that it ever should have meant that anyways. Some big examples of that I'm highlighting here, which are 2 papers by 2 pharmaceutical companies, Bayer and Amgen, and an essay by John Anides, who in 2009 published a still really important paper on why most published research findings are false. So before we get back into these papers a little bit, all of this, everything that I've talked about, that Nature survey, the words that we're using, kind of really hinges on a definition, which is, what do we mean by reproducibility? What do we mean by that? And there's tons of definitions out there, and my own definitions keep changing over time. The more and more I think about it, the more and more I have a harder time with it. And actually, Brian and Nozick, our director here, and I are writing a paper right now on this exact topic once again, only to muddy the waters, I think. But I think I'm gonna take this concept that Steve Goodman and company wrote back in 2016, only because I think it helps illustrate a point that I grapple with a lot, which is it's not a one-size-fits-all, that there are many barriers that one has to think through when they're thinking about the term reproducibility. And I really like the fact that it's broken down first into the methods, right? That you have to be able to have sufficient information to basically be able to redo the experiment if you wanted to first, right? So this methods reproducibility is the ability to implement. Doesn't mean that you will do it. It just means that you should be able to have the necessary information to do it if you wanted to. And again, I can talk about that if you guys ask some questions, or if you want me to kind of get off script a little bit about barriers that we've had right there at that stage in our own project, which is if I don't have access to the same data and information and materials, I can't exactly do it again. So I can't actually ask the second question, which is, essentially a bit of a results reproducibility, or more importantly, inferential reproducibility, which is if I was to be able to run the experiment again, that means if I had enough information, do I get the same results? And then if I get the same results, can I make the same conclusions? And that's actually a really, really important point, because one, there's no clear definition of what it means to get the same result. I'll talk about that in a bit and give you an example of what it means to look at reproducibility in a different way. And then what I really am fascinated with is that I think on many levels, the inferential reproducibility, the conclusions that we draw, are actually in some ways still the best way to ask that simple, relatively simple question: is it a reproducible result or not? What I mean by that is that when you get back to the survey, if you ask a whole bunch of people, is this reproducible or not, you're probably going to get a whole bunch of different answers. And so everybody might not be talking about the same thing or having the exact same definition. What we never really get into directly in any of the projects we're doing, including the reproducibility project in cancer biology, is I think what some people refer to as reproducibility, such as robustness or generalizability. And that's tough, because in theory, that's what research is doing at large, right? When I publish a finding in a given mouse model, let's say, I want to say, does it generalize out beyond that mouse model, right? That's why we have a preclinical to clinical pipeline, for instance. It's because we're constantly trying to ask the questions of, is it robust and is it generalizable? Because that's essentially where we get to applicability. And those are really hard ones to address, but I think they're actually really important to get at, because in many ways, that's actually the heart of the credibility of research at large. And I think that's what, with the Brazilian Reproducibility Initiative, that you guys are actually trying to step into those waters, which are really tough to address. So I appreciate that. I'm going to add one more thing just to muddy the waters a bit more on replication and reproducibility, which is, it's not just as simple as saying, oh, I'm going to replicate and do the same experiment. And the reason why is because there are some differences, and I'm going to kind of bin them into two categories, what we'll call direct replication, what we'll call conceptual replication. So if I'm doing a direct replication, this is what we were trying to do in the cancer biology project. I'm using the exact same procedure, not changing anything. Versus, I think what we do relatively well in the literature, which is I'll do a conceptual replication. I'll change the procedure on purpose. So an example of that would be I'm looking at protein phosphorylation. Maybe in a direct replication, it was done with Western blot, but maybe I can completely change the procedure and do it by mass spec. I also, when I'm doing a direct replication, I'm testing the assumptions to produce a finding. But I'm testing what is thought to be necessary to get the same result. Versus a conceptual replication, which I'm just testing that same hypothesis. I'm asking, the same question, but using a different procedure to get at it. And there's an important difference there, because if I'm doing a direct replication, I'm establishing that the finding, the original finding, I'm asking whether it's reproducible or not, because I'm doing the exact same test, and I'm able to ask that very simple question. In the conceptual replication, we're asking a different question, right? We're asking about converging evidence to explain a finding. And that's important because what that means is the direct and conceptual cannot do the opposite. So a direct replication, if it can establish that a finding is reproducible, does not mean that it's valid, because I could make the same mistake again, right? If the model that I'm using, the mouse model I'm using, has some, essentially unknown factor that's causing an effect, I'll just repeat that again when I use the exact same mouse model. In a conceptual application, even though I can have two different mouse models, which will kind of get around that issue that I just explained, I could get those both wrong. So I really need both of these to be able to have a better idea of, am I accurately building knowledge? So I raised this because we very much in our project are focusing on the direct replication side of things. All right, so we're gonna go back to these papers, these kind of really earth-shattering papers, especially, when these came out, these very much, I got very concerned on this, but I'm gonna raise a couple things, because this actually essentially illustrates why we started the project. And that's because there's some caveats to those papers. John Aniti's paper about why most research findings are false, it was a simulation, right? It was based off the characteristics of the published literature. It was not a meta-analysis of the literature itself. So it's a simulation of what we would expect to find. Similarly, the Bayer and Amgen studies were correspondences or comments. They were white papers. They told us that they tried to do something and they found some result, but we never knew what papers they were trying to replicate. They never reported the results, never reported any of the methodology that they used to assess if something was the same or not. And I think these are really important, because what that meant is that they raised the alarm that something should be talked about. But I think what it meant is that it kind of gave the very next step, which is what we took, which is we should do this in the open and have a conversation about it. And that's essentially what we started back in 2013. It feels so long ago, which is to start the Reproducibility Project in cancer biology. So as I mentioned, it's being funded by the Arnold Foundation. And this is not just the Center for Open Science working by ourselves. We needed a network of labs to be able to conduct these researchers, and our partners on the project is Science Exchange, which is based in Palo Alto, California in the US. And they essentially provide us a network of labs that we've been using to essentially source all the experiments to. And importantly, I think, even if it's taken us a bit longer to finish everything, but I think it's created that dialogue, which is one of our main intents with this project, is our publisher platform is the journal eLife. eLife's an open source, open access publisher, which is really aligned with the work that we're doing, and we've been publishing everything through them for this project. All right, so let me give you a couple more characteristics about RPCB, and then I'm going to pause, and I'm going to kind of give you a rundown a bit of our scope, and then I'm going to pause and take some questions in a couple slides. So some characteristics of the study for those who maybe don't know anything about us. So we, back in 2013, started the project. We focused in on preclinical cancer studies that were published. We did this by essentially, at the time in 2013, looking at the literature during the years 2010, 2011, 2012, and trying to find high impact papers. We chose high impact basically because it was the ones making the most noise, if you want to use that. But we defined it as citations and altmetric scores. So, basically how many times something was being downloaded, how much tweets were happening around it, basically how much attention it was getting at the time. And then we ranked them accordingly and just picked the top papers from each of those 3 years. Again, excluding anything that did not include experiments that we could replicate and source to our network, or that weren't experimental results that we were focusing on. So clinical trials, for instance, would be out of the equation, or some emerging technique would be out of the equation. We also did not focus on any mass spectrometry or proteomics work, just because, or genetics work. That's just kind of a whole nother beast. But we wouldn't keep those papers in if they included a couple experiments that were either, kind of cell biology, cancer biology experiments that were a little more doable in terms of our framework. And the reason why is because we were not replicating every experiment from every paper. We were only, replicating 2 to 3 per paper on average. We also, back in 2013, because we were doing such an open project, we said, this is how many papers we're going to do. And we said the number was 50. I forget where that number came from. And over time, we've reduced that down to 29. Right now, we have 13 fully published results to date, and we're going to be finishing up the rest. The experimental work is done. But we know that not all of the 29 that we started are finished completely. And there's a variety of reasons for that that I can get into in later slides. As I mentioned, one of the keys to what we were trying to do is that we wanted to make this as openly as possible. So how we selected our papers, all the methods, all the data, all the analysis, we make completely openly available, and we're using the Open Science Framework for this. So that's our GUID for the project that we've been slowly making more and more open over time as we've been publishing papers. I think one important thing that we were never intending to do with this project, and I think no paper can really do this, it's not meant to say that we can prove or disprove the original studies, right? Each one's a single study, and so we went from an N of 1 to an N of 2, and we still should not be drawing strong conclusions from that. So that doesn't mean that you can dismiss either one, but it also means that that's not our intent. So the word validate I have a tough time with, because it's really hard to validate a study. I can validate a reagent, but it's really hard to validate a study. I think only time and experimentation can do that over a period of time. So I'm going to introduce one more concept before I kind of give you a big overview of the project and pause for questions. One of the things that we were doing that's different from your project that you're getting involved in is the way that we are choosing to publish each of our studies, each of our replication studies. And so I'm going to introduce a concept for those that aren't familiar with it called registered reports. So if we first think about the traditional academic publishing model, any journal in a very simplistic manner, it basically follows a trajectory that you see here, which is I develop some idea, I get a grant, or I get funding, or I use funding to design a study, and then I collect and analyze my data. Eventually I get to the time when I write my report, and when I decide to, and if I'm lucky enough, I get that to undergo peer review before it gets published. So importantly, peer review occurs at one time, after I've done all the analysis, after I've decided to put what I do and do not want into my report. And it's only at that time that it gets peer reviewed. So it gets peer reviewed not just on the design of the study, but also on the outcomes of the study. In the registered reports model, which is what we're using in the cancer biology project with eLife, we do 2 stages of peer review. One, right at the time of designing the study, and then again, after we've completed the analysis and written up the paper. And the reason for that is because, one, it allows us to get feedback at a really crucial time, which is right when we design the study. When we conduct a replication, we want to get as much feedback as we can before we start it, and I'll show that in the next slide. The other advantage is when we get in principle acceptance at stage 1, when eLife agrees that, yes, we did a good job designing this replication, they agree to publish our outcome no matter what it is. And I think that's really important because we don't see many replications being conducted or published. And a big reason for that is you either, 1, maybe found a very similar finding and it's hard to publish, or 2, you found a negative finding, something that was counter to the original study and it's hard to publish. So this kind of gets away from that publication bias and allows us to kind of increase the credibility of our results as well. All right, last slide, I'm going to take some questions. So this is an overview that's still accurate in terms of the way that our process has unfolded. And I think this is a good a way to kind of describe our process a little bit in a simplified manner. So when we identified the papers that we were going to do, the very first thing we did, like I mentioned, was we're just going to look for a couple experiments. And this was done slowly, actually, over time with getting a number of inputs from people on the team, and including even input from the original authors, which is, well, what were the key conclusions from the paper, and can we identify a subset of experiments that we're going to replicate? Once we identify a subset of those, then the very first thing we started with was the paper, right? It's a natural place to start. So we obtained everything that we could from the key figures in the paper. So we annotated the papers, figured out, what we knew, and importantly, what we didn't know. And actually that illustration down there of the yellow coded, I'll show you a slide after this, after we take some questions, that looks just like that, because it's exactly what we did, is we tried to figure out everything that we could from the paper, or that was referenced in the paper. After we did that, we would then also ask the authors for information, right? We didn't just ask for commentary from, ourselves looking at the paper. We actually went and reached out to the original authors and said, hey, here's our protocol. Can you help us fill in the missing holes? And whether we were able to get in touch with them or not, and that's a variable that we keep track of, we would shift over and eventually source this to the labs. This is where I said that our partner Science Exchange come in. We would use contract labs through the Science Exchange network, and we'd work with them to say, great, well, either this is what the authors told us, can you do it? Or there are still some holes that are missing, can you fill them in? What are you actually going to do, right? Let's just not leave it a hole. Let's actually describe as much detail as we can up front. And this doesn't just include the protocols, it also includes the sample size that we're going to use, the analysis strategy that we're going to use. So this gives as much of a full understanding of what we're going to do before we do it. So that way we have no reason to think that it's not going to succeed. And then on top of that, we then sent that using the registered reports model to eLife. So that way we'd have another opportunity for others to weigh in. So again, and eLife in a formal peer review process, the original study author would be invited to come back, a statistician would be invited to review our work, and other experts in the field would be invited to review our work. And at this point, they're only reviewing our methodology. They're reviewing what we're planning to do, not what we found. And after finally completing all of those steps, which is actually really great because it allowed to give us a lot of information beforehand, but it also kind of gave us a really good faith approach before we actually embarked on the research. That's when we performed the experiments. And so that's what we've been doing for the past couple of years, is finishing up this process to get as many of these through that we could, and then performing the experiments. And I'll talk about it a little bit as we go forward, but even after all of this work of identifying what's in the paper, talking to the authors, and getting peer review, we still had a lot of experiments where things would not work according to how they were described. And some of those we're able to publish, and you can read some of those in our papers, and other ones, it's actually a reason why we're not able to finish all of our studies. Or that's a reason why we had to cut back on the number of studies, that we've had to invest more resources in fewer experiments because it just became more costly to do that. And as I mentioned, because we have in principle acceptance, we slowly then, and this is kind of where we're at right now, which is publishing our findings, going through that final peer review, making sure that we're presenting everything in a clear way, and then having that be accepted and published on eLife. So I'm going to pause here if that's okay, because I think it's been about 15 minutes, 20 minutes, yeah. And just kind of see if there's any questions in our general process or anything that I've talked about.
Well, if anybody has questions, you can either raise your hand and we'll put you online, or you can write them, send a written question as well. So I think Juliana is keeping track of that.
Yeah.
Do we have? I have questions, but I can wait for other people first. But if nobody comes up, I'll take the lead. Two quick questions. Maybe I have asked you already, but like, do you have numbers for author engagement? How many of the original authors actually respond, and how helpful they are? And,
yeah.
And second thing, how long did the whole protocol thing take you, more or less? I mean, just trying to figure out if we're slow, or if, or how much we have to, like, from selecting the paper until being ready to start. Like, how long a process was that?
Yeah, those are great questions. Those are really good questions. So I'll start with the first one you asked about author engagement. It's actually just looking at it today. So it has different steps in terms of where the engagement would occur. We'd be contacting them right off the bat. And so right off the bat, we had about 50% engagement of authors, but then we'd continue to try as we were developing the protocol. So we never really kind of gave up until we essentially decided that we had a protocol that we were getting close to submit, and then we were just going to stop contacting them. And so in the end, we had about 2/3 engagement, which is a lot less than what we had in the psychology project, where it was about, I think, 95% engagement with original authors. However,
what was the other third? People just didn't respond, or did they say we're not going to help, or?
Yeah, that's a good one. It's a mix. So we've had a couple that most of them would not respond. So we would try to go through, and this was not just going with the corresponding author. We would actually try to be careful and kind of navigate to the first author as well if we could. So a couple of times it was just couldn't get any contact. So we essentially had to just move on. We did have a couple times where somebody just basically said, no, I'm not going to help with your project. One time for I think for very interesting reasons, which is I think it's better that you do your project with just the paper and not engagement with me as an author. And I actually think that's really cool because there's nothing saying that one has to engage the author. There's obviously advantages to that, but we shouldn't be relying upon it, at least in my opinion. So yeah, it's a healthy mix of what we found. We also found sometimes where, when I say 2/3 engagement too, we'd have some where they would barely, this is any engagement, by the way, right? This just means we've gotten some communication. There were some times where we'd get next to no communication in our informal process, but then we knew it was the original author in the formal review of the registered report because they would disclose their name to eLife during peer review. So it was interesting that sometimes it just, for whatever reason, they would not engage with us, but they engaged during the formal peer review. So there's an advantage of that, which is we actually got some feedback. And even from the authors that we informally, sometimes, I think that different review caused them to maybe surface a couple more comments or some corrections in our process, which was really helpful. So yeah, so not 100% and not near it, but more than 50, right? So then the process, protocol process. So yeah, we have it broken down in terms of time, and sadly it's the whole, because we keep track of the time from when we start something all the way to when we're finishing things. And obviously there's a lot of variables at play. So I know that doesn't really help your answer, but whenever I give a rough estimate of what we're doing, it's because even though we're tracking it individual, because they're all occurring at the same time, they're all dependent upon each other. So we have to keep that in mind. But if we just take like an average paper and we assume that we have, in our case, we have author engagement, or we have clear delineation that the author did not engage. The reason I say that is because sometimes we would have it where it was slow trickle information from the authors. And so instead of us imposing a deadline saying, you've got 1 month to give us all your info, we just kept working with them slowly. So to get to the point where we're able to contract the labs, it could take anywhere from, just a couple weeks, to be quite honest with you, depending on the nature of what was in the paper, to months. And that's mostly because of two big reasons. One is if there was a lot of information missing from the methods, there was a lot of back and forth with the labs to make sure that we were on a level playing field in terms of what we were going to assume. But the two, it would also get long if we had engagement from the authors, because then there'd be this back and forth, because you can never do the same thing twice, right? We would have to change something. So it'd be, let's say I'm going to use a different program to analyze your data, or I'm going to use a different antibody. Let's say it's not available. There's a ton of back and forth in terms of, is that appropriate or not? And so that would kind of slow us down a little bit, because again, go back to what our approach was. We didn't want to have any reason for this not to succeed. So we wanted to cross all of those off before we submitted any paper. And then the peer review itself, that's pretty classic in terms of how long it would take for eLife. They're pretty quick, it'd be about a month for each round, and it'd be very quick in between us. So 2-3 months, if not a little bit longer, but usually about 3 months it would take before we went from submission to acceptance of a registered report. So that gives you a pretty wide time frame on average of probably somewhere in that 6-month range just to get the paper accepted. But again, those were all moving at the same time, right? We didn't submit all at the same time, so that took a couple years for us finally to finish with 29, which is when we decided to stop it.
More questions?
We don't have questions, Olavo.
So I have a third one. I mean, you mentioned the source of the antibody. So what was your approach there? I mean, were you looking to just repeat every manufacturer exactly as it was? Or, I mean, did you have like a relaxed thing, like, okay, maybe just your regular, I mean, very common reagents could be exchanged? Or, I mean, how did you work that out? Because, I mean, that's one big thing for us. I mean, should we keep the same manufacturer? Can we change? Should we decide what can be changed, what cannot? So what was your approach in terms of reagents?
Yeah, so I'll tell you my approach, and then I'll tell you what I'd recommend, because they're not the same, not entirely the same. So our approach on this one was, because we were looking from the project from two perspectives. One was similar to, I think, what you're getting at, which is I just want to see the result. I want to try a good faith replication. The other one that we're trying to do is actually to understand, and I'll show you a slide of work that we basically were building off of, which is I want to know if I can get the same reagent as you. I want to know if I understand what you used. So that way, if I'm changing it, I'm doing it on purpose versus I don't know what you actually did. So our attempt was always to source everything identical, focusing always on the key reagents, right? Models, antibodies, drugs, and the such, cell lines. So we'd always try to do the same thing, and we'd even ask them if they had something that was unique to share, but we'd always keep track of it only because we were very curious in the question of, did it matter, right? Does it matter if I change the source? And we knew that we couldn't answer that question unless we tracked it. And so that's why we did it. It's kind of those two elements, because they're intertwined with each other. In hindsight, I think what we should have done is focused very heavily on a quick assessment of what was known in the papers, focusing completely on just key elements, actually asking what those key elements are, or assessing what those key elements are, versus what we were doing, which was kind of trying to be as inclusive as possible, making basically kind of open-ended judgment calls on, okay, if it's not described in the paper sufficiently, like, we will use, basically we'll have an external person accept what that alternative would be. The reason that I think that coming up with a process to at least identify, of the key reagents, which ones do I think I can change, and not worry about the rest, it just means that you can have more of a streamlined process of it. I think the risk you have is you don't kind of know what is key. I'll give you an example. We published a paper, one of our, I think it's like the 6th or 7th replication study we published. And element of this was the way that the animals were going to be prepared for a tumor injection. So the original study used radiation, and in ours, we were going to use a drug treatment, right, to basically prepare the cells to inject them with leukemia cells. So maybe I'd count that as a key reagent. I don't know. It's kind of hard to tell, right, because it's so close. And the reason we did it differently is because the lab that we had sourced it to did not have the radiation instrumentation to do this, which it just doesn't surprise me, since I know there's a lot of movement, at least in the US, to get away from that. And so, we surfaced that. We did everything else the same. We get the results back, and we don't have a similar result. And the reason for the big reason that kind of came up in peer review was, well, it's probably because you guys changed the way that you prep the animals for engraftment, right? So now you get to this point of, we know there's a difference because we changed it and we kept track of it. But is that something that you guys are going to, or anybody want to consider? Is it important or not? So I think, the question of keeping track of kind of key characteristics and moving them, but always keeping track of when and how you move them, is actually key, because it starts to get at that generalizability and really more robustness question. Because you would hope that you're robust to it, but you can't ask the question if you're robust until you actually know what you're changing. And so in the ideal world, what we would have done is we would have done the experiment the exact same way, using the exact same reagents, and we would have done the experiment changing the reagents. To ask that question doesn't matter, but we just didn't have the luxury, so the two are intertwined. And I think you're going to have the same problem, where if you end up having them and you change them, there's no reason not to change them. They're very valid, but it's also going to be the very first thing that gets flagged why they're not the same. But again, depending on your purposes, if you're saying, I want to make sure I'm robust to changes in antibody supplier, but as long as this antibody is validated for the technique I'm using, and is indeed validated for the target that it identifies, it should be fair game, right, in theory.
Yeah. So we have a couple of questions, written questions. I don't know if people want to show up. On the video. If not, you can just read it. So one is, when you say that you contact authors to get them to fill in the holes, you're saying that articles do not offer info enough in order to guarantee that a peer can reproduce in experiments. What can you say about improving documentation practices in labs and the idea of sharing open notebooks?
I'm going to answer that question in the rest of my slides. So that's a great question. That's the heart of it, I think. And I'm going to talk about that in the rest of my slides. I'm going to defer if that's okay.
I think it is. And second one, can you share your experience on using same animal cell strains, but from different sources?
Yeah.
So what is the impact of reproducibility? What about across different countries, animal facilities, etc.?
Yeah, so, I'm glad that you bring that up. So, right, animal models and cell line themselves. So I'm gonna answer it a bit in a question I got asked by a reporter that's very similar to what you guys just asked now, which is, do you have to use the same thing, or are you going to change it? And the example here is, well, if you get the cell line, and it could have been commercially available or through a repository like ATCC or another one like that, but you choose instead to get it from the original lab, and it turns out it's contaminated, with, say, mycoplasma or something, are you just going to use that or no? And this was a question being asked to me from a reporter, and I was like, and it got asked to me at the exact same time. We actually had a very similar case occur with an animal, where we got the animal from the original lab that did this. They sent it from Europe to the US lab that was conducting the replication. It gets to the lab and it's contaminated with lots of pathogens. And so we're confronted right off the bat with, well, what should we do? Because apparently that's what they used, is contaminated cell lines and contaminated mouse models. But we obviously did not follow through with that. We were like, well, no, we can't do that. One, because the animal facility would not allow it in. It had to be in quarantine. And then in the cell line, we always were checking for that because we did not want that, because we know that it has problems. But you get confronted, I think, with this real big issue, which is you do need to set some QC standards of what you want to impose on your project. So for us, we always ensured that the animals would be checked for pathogens, the cell lines would be checked for authentication, to ensure that they're actually what we think they are, and that will ensure that they're not contaminated themselves. And if they were, we were going to either resource them out or have to clean them up depending on the situation that we got into. And surprisingly, we did find this happen, like I mentioned, both with animals and in cell lines. Actually, the paper that we just published, the most recent replication study we just published, we were just talking about this beforehand, is a good illustration of that exact issue, where we got the cell lines from the original authors. This is a xenograft model, basically kept in suspension, gets to the lab that's conducting the research. This lab happens to be in China, in Beijing this time. Basically, they take it, they clean it the way they were supposed to, get them ready for injection. We segment some off for authentication and contamination checks, while the rest get injected into the animal to continue the growth. These are prostate cancer cells. Like I said, this is a paper we just published. The tumor doesn't grow. We look back at the STR profile, and it turns out that it's basically XX, not XY. We go back to the authors that originally identified the cells many years ago, ask them for a sample, we get it from them, it comes out exactly what it's supposed to be, the tumors start growing, and then you can look at the paper to see what the results are, but we basically didn't find any effects, unlike the original study, which found effects for what they were studying. So I think that's a really important question, because whether you're getting it commercially, whether you're getting it from another lab source, you're going to inevitably run into this issue of, are you sure you're getting what you think you are? And what are the QC standards you want on it? Because you're going to get confronted on something. And this is, I think, the key of it, which is cell lines and animal models are definitely the heart of almost all the research in biomedical landscape. So that's a good question. I don't know if I answered that sufficiently too. There's a lot more to go in there, so happy to expand.
Yeah, but just to understand, I mean, was your original approach to go for the same source, or did you, I mean, just get cells, for example, from commercial
the goal was to go for the same source at all costs, but we would change if we had to, right? So this was an example of if we could get it from the same source, that was the intent, always the same source. And if we couldn't get the same source, then we would switch it to the same source, but from a different location. So we would hunt down labs sometimes if it wasn't in a repository. Thankfully, a lot of the cell lines and animal models we were using were in repositories that were openly available to anybody. But if we couldn't find them, we basically would look in the literature to say, hey, can somebody else who's used this before give it to them? And so that does create more work, right? Because we're trying to use the same cell line, but we're not using it from the same source per se, because the source is not available anymore. We would change the source. So you're going to get into MTAs here too, by the way, when you start to get into this, because like some of the stuff you're going to hit into, it's not openly available. So you're going to have to probably, navigate material transfer agreements. Just as an FYI, we definitely ran into that, and it can cause headaches if you're not ready for it. So just be prepared, like depending on what your institution is and the policies are, that if you need an MTA to go through, be prepared to try to identify those up front or try to avoid them if they're going to be in the way.
Anyone else? More questions? Otherwise, I think you're ready to continue.
Cool. So I'm going to
questions.
the first question. Oh. Are we all set? Okay. All right. So there's a great question that was asking about, just the information we got from papers and how we prepare ourselves. And it's actually kind of where I wanted to go a little bit and to touch on two of the points that I spoke about earlier. So I'm going to give you a rundown of what we essentially were finding from the publication and from our process. Again, I'm doing a more qualitative approach here. We are summarizing things in a more quantitative manner, but I'm not going to talk about that at this time. You guys are going to have to wait until I finish all that up and write our paper that hopefully we'll be submitting later this year. So here's just an example of a paper where, you know, this is the experiment that we were going to replicate, science paper. What we've got is what's written in the short text, what's written in the figure legends and in the figures itself. And then, you know, we get to the material methods, and we would not surprisingly find situations where they'd say, if you want to know the method, go to another paper. And then of course we'd follow that paper and it'd say, go to another paper. And eventually you go somewhere, you follow the literature trail, and it takes you to nothing. So you're left with no methods. And we would have that where sometimes there literally would be no method stated whatsoever. It was only stated as described elsewhere or following standard procedures, whatever that means. And so what we would do, just like I was showing in the slide that somebody summarized for us before, is we would write out step-by-step instructions, and then we'd highlight in yellow what we didn't know. That includes the materials that we were focused on. It includes the methodology. It includes the methodology that we knew should exist, even though it was not stated explicitly. And that would happen quite a bit of times where we knew that there would be a step in the process that was being, you know, essentially cut out, and that we needed information because an assumption was going to be made. And so we keep track of this all the time for each of our papers, and we essentially have the first version of this from the paper, and then we would just basically version that over time as we got information from the authors and the labs. So let me give you some ideas of, like, you know, simple or big problems, but relatively simple questions from the papers. We wouldn't know the antibodies that were being used. Maybe we'd say, oh, it's a MIC antibody. Well, there's, you know, thousands of MIC antibodies out there, hundreds at least. Which one was it? Right? What sex were the mice? We didn't know that sometimes. We had no idea what the sex of the mice were. We didn't know how long treatment periods were for. We didn't know what instrument software was being used, let alone version control. There were some protocols, like I said, that were just completely missing, right? There was one time where we had nothing on a staining protocol. So we had to assume it. Even the way the variables are being defined, right? We had one good example where it said, you know, we found an increase in the positive area of staining. Well, great. How did you define positive area, right? How did you measure that? Because we had no idea how it was. Now I could come up with an idea, but that means that we might not be measuring the exact same thing. And of course, a common undertone of the whole aspect, because we wanted to make sure that our own experiments were high powered, we wanted the data. We wanted to know what the original result was, and how to best design our experiment. And that was always a constant aspect, just like the methods, which is we didn't really have the information from the publication. Again, here's some responses, you know, not obviously tied to anybody, so you can't really figure this out, but from the authors, we'd get responses that I think really hits at the head of what we're not prepared for to deal with this. And it gets back to the point that we're busy and we're not actually setting up to expose more information in the papers themselves. So, you know, a lot of times the information would get lost when they had to edit the text down to size, right? A lot of the papers that we focused on are science, nature, and cell papers, just because those happen to be the ones that came through our search criteria, and those also, especially science and nature, have really short text limits. And so even though we still had the supplemental materials to get access to, there was just not a lot of information in the text. And part of that is an editorial process that could change. And that, you know, that's why our policy team is here. The data would be missing, right? And sometimes they, you know, either couldn't do it, they'd have to go digging in their backups to try to find a file that they have no idea where it is, or we definitely had somewhere the data were gone. And they, you know, disclosed that for us that, yeah, that, you know, I moved and that hard drive is gone now. So I don't have the data with me and I can't get the data anymore. Same thing with the protocol documents, right? We would share the document with us, ask them for feedback. And the problem is it was also done by different people that have all left, right? A project kind of done at a moment in time. And the individuals who were, you know, crucial to that project probably were grad students or postdocs at the time that are now off somewhere else. And it's really hard to basically get everybody back to say, what did you actually do if it's not in the paper? Like I said, you know, raw data is missing, and it's just not really accessible anymore. These details, you know, even though at this time we're only, you know, a couple years out, right? 3-4 years max at the time. It's pretty much once it got published, I think there's nothing set to kind of share it and move it forward. So I'll talk about it a bit on kind of ways to move that forward and what we're trying to practice in our own project, and I'd recommend you guys doing. So what are the other roadblocks that we've had to reproducibility? So a big thing that we have is representative images. There's no error reporting, right? We'd have a single image from a Western blot or an IF image. So you have to think about that. What do you do? What do you do when you get one image? How do you know if you get the same result or not? It's subjective, right? It's, okay, what do you think? Does it look the same? And then the question is it truly representative? I don't really know. I don't have all the original images. I only have one. So I have to just kind of go off of that. we'd have constant incorrect reporting of statistics if there were statistics at all included. We wouldn't know what the groups were being compared. It was not, sometimes it was easy to tell, but there were a lot of times when we had no idea which groups they were actually comparing or what type of tests they were doing. Like I mentioned before, we couldn't get data or we couldn't get methodological details. I couldn't find it, even giving them weeks to dig through lab notebooks. Again, we tried to not have time be a barrier within reason. And again, in hindsight, you know, we could have put some breaks on that, but that's also a big part of our project, is to figure out where are the challenges. Like I said, methodological details are missing. We didn't really understand all the information. And we would miss these, like, we were talking about this before in the questions, these minimal standards for quality control checks, right? We didn't understand what exactly was being done at the time. Like, take the example of the STR profile of a cell line. I don't know what it was in the original paper, right? Unless you share it or you point to a database, I have no idea what I'm comparing to. So when I change something, I don't know if we're on the same bar. And the same thing with reagents, right? If I'm going to change it, what's the minimal information for quality control checks for an antibody that I should try to mirror if I change the antibody? And I think that's an important thing to think about, which is kind of what are those benchmarks? What are those minimum information standards that you need to set, and how similar are they to the original? All right, as I mentioned, the other big question we have that we're still facing right now that you're going to have is how you assess reproducibility, how you assess if something's the same. And there's a lot of different approaches that are out there that we've published on. I'll give you an example of one from psychology. I think I maybe cut out from my own, which is you could just do statistical significance, the effect size, right? You can do meta-analytic approaches to combine them. You can ask people, what do you think, right? Did I get the same answer or not? And of course, what's behind all of this is not just the outcome that you get, but all those slight differences that you have between them, right? If it turns out I did something slightly different than yours, does that change your interpretation of the data? So I think assessing reproducibility is something we need to continually go at. And I think it's exciting to see what you come out with your project, because I think this is, you're going to add a lot more into here. And that's a really crucial element to do, is to kind of assess this. And there's a real crucial point, which is the best time to figure out what you want to do is before you know your results, because then you're not biased about those results. So an example, as I mentioned, about looking at psychology, and I had put in here a kind of a pilot, and I decided to take it out, which we're doing the same approaches in the cancer biology work, and we've got some preliminary data. But here's just two elements that we can look at between the original and the replication studies. On the left there, an A is looking at just p-values. So the original study, there were 97% of the original studies had a p-value less than 0.05, that magical number. And the replication, it was 37%. So that's one of the numbers that were used when this paper was being reported. And we'll have our own number for cancer biology, and I'm sure you'll have your own too. But that's not the only way to assess if something replicated or not. Another one is B, is looking at the effect size. So if we make all of the studies have a common effect size, and in the original study, we treat them all as one direction as positive, you see that there's this, you know, basically mean and median effect size that's sitting around, you know, 0.37, 0.35, and that you get regression to the mean, right? You see a decrease in the average and median effect size in the replication. So not only do you get less significant results, but you get a less precise estimate. You basically see a decrease in the effect size, the actual difference between the conditions. So these are two big measures that I think are really helpful for us to kind of tackle that question. And like I said, the other big one is just interpretation. What do individuals think? So yeah, we'll have more of this. All right, so there's another question in terms of just thinking about there's a lot of issues. I could go on and on and on about this. I just want to highlight a couple big ones, and these are just looking back at the way we currently conduct research and the way we currently kind of present our research. And I talked about a lot of these, disorganization and loss of materials and data, but there's some of these that I have not, and I'm sure you can read more or you know more about. What's interesting that I want to raise is just that, especially when we think of analysis and nulls and lack of replication, these are not new issues. These have been talked about since the 60s. Back when Tukey was talking about them from the statistician point of view. But I do want to talk a little bit about, I think, some of the issues that we can fix today, which is just that disorganization and sharing of information, the one that I highlighted before and the one that was the question. So I'll give you two more examples of kind of why these are important, why we should be doing them and preparing for them now before we publish, opposed to trying to figure it out after we publish. One is unique identification of antibodies. So this is work from 2013 from the Resource Identification Initiative, They also are the ones behind doing RRIDs. If you don't know what those are, I highly recommend looking at them, but they're basically unique identifiers for key reagents, such as cell line and antibodies. And this is a paper they published where they were looking through the biomedical literature to ask, well, how complete are certain key elements when I look across different journal articles and basically that are covering across different disciplines. And what you see is that there are some reagents, such as knockdown reagents or the actual model organism, that are pretty good, although they vary quite a bit across disciplines. Some of these are really horrible, like the actual DNA construct being described sufficiently. You know, maybe 25% of the literature describes that from this study that was done in 2013. And so I think, you know, that's something to really keep track of, especially the fact that it changes across discipline, which is you might not have the information in the materials presented in the paper to be able to do it sufficiently. And look at antibodies as a great example. So we're going to be doing something very similar to this for the cancer biology work. We've been annotating all of the work that we could gather from the original papers, and also trying to figure out not just what wasn't reported, but also, and this is again looking at what wasn't reported sufficiently. It tells you that they knew there was an antibody, but there's not enough information to know what antibody, what source and catalog number. We also have additional information, which is sometimes we knew that there was an antibody, but that was never described. And so basically everything here that we're looking at on this slide is essentially a good faith estimate, right? It's the highest estimate we can get, because the reality is it's lower. That when you actually try to do the empirical work, you'll realize that there's more information missing than not described sufficiently. Another example of this is just looking at data. There's been a number of papers here that have looked at this. Most recent one by Victoria Stodden's group that was published in PNAS, basically trying to get data from previously published papers. This graph you see here is from an article from a handful of years ago from Tim Vine. And they basically found that, you know, within 5 years, the ability to extract data from a paper just tanks really fast. It rapidly declines over age. So we see here in the graph, the red dots are some individual time points, looking at hundreds of articles from the time they conducted their study, from 1 year out to about 20 years out. And the farther out you go, the harder it is to get the data from that underlies the paper. What that means is they have a hard time accessing the data and code from the original authors. And this is similar to what Victoria Stodden's paper is from PNAS that was published last year, where they only found 44% of data and code were available in the hundreds of papers that they looked. And of that, only 26% of the total could be reproduced. That means I could run the same code on the same data and get the same result. This isn't redoing the experiment, right? This is computational reproducibility. So this information or lack of information is a big barrier, I think, in terms of our understanding of the literature. So part of that is because it's hard to manage our research, as we know, right? Collaboration, accessibility, organization, trying to do all this, tracking versions, and we're doing it as humans. So we're prone to error. And part of the error is because we're humans, not because we're intending per se to misinformation. It's because we don't know. We're more busy trying to get the research done than we are about documenting our process along the way. And I think that's going to be the key to moving forward, which is not trying to figure out what should I put in the paper, but trying to make it so that you can expose things when you publish your paper. And I'll give you an example of what we're doing in the cancer bio work. But first, I just want to give you a nice, there's one more quote I'll give you at the end that I think summarizes this well, which is the effort that goes into this is not so much for the field at large. It is, but the best person who's going to succeed is yourself. It's your future self. And this is where a lot of librarians will come in, and I completely agree with this. All right, so let me give you an example of what we're using. So we're using OSF, it's a platform that we develop here to help with collaboration and sharing of data. You guys are having a project yourself on this. So whenever we publish a paper, we have an OSF project with this. So this is just a short screenshot of one of our papers that we published. In here, we've got the title of the paper, and these components here on the right side are each of the experiments that we're doing. And as you can see there, kind of in the bottom left, it has basically its file management, right? That we have image files and protocol files. We just basically keep track of this and communicate with all of the different authors that are a part of this project. So what that means is because we're doing that the whole time privately, when it comes time to publish, all we have to do is make that open and then cite it in the paper. So when we write our own methods, we still write a summary of methods, but then we link to the full methods on OSF. Right? So I think this is a path forward, which is if we can preserve information and expose what we were doing at the time we did the experiment, we don't have to worry about writing it up again. I can just write a brief summary and then point to you where the actual protocol is that I followed, the actual details that I followed. Same thing when we think about images, right? I don't have to worry about, putting all the images in the paper because that's a lot of burden. I can just expose all my images. I can expose all the original raw data counts. I can basically populate links of individual files as I see fit. Same thing with my statistical analysis. I can show you my scripts, right? I can show you the packages I've used, the exact approaches that I was doing, I can expose to you. So then when I publish a paper, I can still, on the left here, you see my paper. We have bar graphs, which I don't really like, but I'll tell you why if you're curious why we did it. We have representative images, B, C, D, E, F, G, just like we do in our typical papers. But then in my figure legend, I can point to you where all the information is on OSF. And if you go to the OSF on the right, you can see all of our images, the hundreds of images that were taken. So that way, when I show you one image and I show you the data that I got from that, somebody can go back and do it again themselves, or they can reanalyze it in a new method if they don't want to follow the method that we chose. And I think this is a path forward to try to help make it easier so that others can build on our work. All right, so I'm going to end with this and open up to questions again, which is just really trying to think about something you can do in your project, because it's going to be a barrier to your project, is trying to think about the article as more than just an article, right? That's just the advertisement for it. That the actual scholarship, what you guys are embarking on now, that's the research. And so the more that you can document and expose that at the end, I think that's the biggest impact that you're going to have. So if you can leave, I'm hoping that we're doing it. If we can leave a trail of a way that maybe can improve upon it so that others don't have to kind of have the pains that we did, maybe that's a path forward. And so with that, I'll stop with this. You're going to download these slides here at this site. And you can always email me if you have additional questions, but I'll stay on, I guess, for a couple more minutes, right? For more questions. I don't know if it's 4 o'clock or 3 o'clock, but.
Yeah, so if anybody has questions, again, you can either write them and we'll read it out, or you can raise your hand using the raise hand button and we'll put you online. I can start with a couple. One is really practical and kind of technical. I mean, to do the back and forth protocol revision with the authors, did you just use like I saw like an annotated text file with a lot of yellow stuff. I mean, did you just use that, or did you try anything more structured? I mean, we're discussing that. We have different views. I mean, should you use like a Google Forms, like all possible steps and like there, versus should you just like write it out in Word and send a text file? So what do you think?
Yeah, it's a great question. So part of it's going to be on the comfort level of who you're engaging with. We ended up defaulting back into what you saw, which was a Word document that we would annotate. We basically would just use Word functionality using writing our questions down in there, and then basically passing it back and forth using comments. And the reason why is we tried, sliding into like Google Docs and using OSF as a platform to try to open up collaboration. I think what we find is that it's the default to, at least the people that we were engaging with, was to work on a document. So we were able to manage it. It was slower. We'd have to obviously internally make sure that we were keeping track of who was doing what, so that way we weren't crossing it, because we'd have so many versions. But the reason why is because I think there was just a comfort level. It's that comfort level that you have to get back to. So I think Google Forms could work. You just might want to pilot it to see if the comfort level is there with the audience that you're going to be engaging on the authors, because you don't want to isolate it. That's the only question I'd say. It's a good point, though.
Yeah. Well, I mean, we have different views on that. So Kleber wins on this one. I'll be the loser. I was trying to push for forms, but maybe
No, I think it's a good. I think it's good.
Yeah. So anybody else has questions? You guys?
No questions here.
Yeah. Then I'll ask the big one. Well, I mean the more conceptual, larger one. I mean. You've been on this for like, I don't know, 5-6 years or something. You're about to embark on another project. So like, if you had to do something differently, what would you change in your original approach here? I mean, what have you learned? I mean, what would you do differently? I mean, what do you think it might have been better to do otherwise?
Yeah, no, I think that's a great question. So I think if I had to do this over again, I think I'd have to have a better, which I do now, a better appreciation of, and it's going to sound pessimistic what I'm saying, but I don't mean it this way, which is that there's just no information out there, that the paper is kind of worthless. that don't think the answer is in the paper. The answer is probably not in the paper. And because the answer is not in the paper, and because it takes a long time to get it from somebody else, that I think if I was to do this over again, I actually would not ask the same question, because I think the question we were trying to ask is really hard to ask, because we're not sharing enough information that it becomes a burden. And that's why it took us so long. So I think what I would do is I would actually shift it a little closer to what you're doing, but I think I'd ask a slightly different question, which is how much time, or how much money really is the question I would ask, how much resources would I put into something to attempt to achieve a reproducible result? So I'll describe what I would do like that. I would give somebody the paper, I'd pick an experiment out of it. I think it's fine to pick a number of experiments out of a paper. I don't think there's anything wrong with that we were doing. And I would basically just say, listen, this is the general question what you're trying to ask. Write up your protocol, write up how you're going to analyze it, how you're going to, compare this, and what you're going to judge as success. So the reason I'm shifting it is, instead of asking the original authors, what do you think? I'm actually saying, you as a replicator, you tell me what you are going to do. And I don't care, you just describe it. I'll just judge to make sure that it's similar enough. And we can have like a group that would do that, but I want to get you started as quick as possible, because I know you're going to run into problems. And what I would do is I would very much hinge it not just on what you're going to do, but I want the answer of what you deem as successful. What I mean by that is it's basically like an effect size, but it's really trying to say, well, what difference do you see as being quote unquote acceptable? And this is again why you might not want to have one group do it. You might want to have like an informal peer review occurring around it. Although I did like the peer review from eLife. I think the peer review adds a huge element. So whether you do it formally or informally, I would always keep that onto it. But what I would do is I would try to get experiments started faster and not have them be focused on what was done in the original per se, but more about what you are going to do to follow up, to attempt to do your definition of a replication. Get that peer reviewed, get that started. And then once you find those results, whether that's during the execution of it, or whether you execute it and you actually get the results. The reason I say that is because from my own experience in this project, we've not been able to finish everything because there are unknown hurdles that we are introduced to. Things don't always work out the way that we plan them to. So we have to put some stopping criteria in there. So basically, I see that as a money-time issue. How much time and how much resources are you going to invest before I tell you stop? And then when you stop, what we'll do is we'll try it one more time. You'll do the same exercise again. You'll say, modify your protocol based on what you know and try it again. And then you'll do that one more time, and then we'll assess that at the end. And the reason I say that is because I've realized in this project that, one, our papers don't give us enough information, so let's stop going off of them. Let's go off our interpretation, which is a bit pessimistic, I know. Let's keep the peer review and state what you're going to do up front, because that's what's really been the most amazing thing on this, which is it's trying to state what you're going to do and why you're doing it is a really important exercise. And it's really important for a replication project, just like an original project. But that we shouldn't expect, because it's so new and because we lack information, that we're going to get it done on the first try, that we should give it 2 tries. And the reason I would put a break on 2 is only because it's so resource intense. Maybe you could argue for 3, but I do think more than 1 is worth it. But then you have to stop it, because the question is, how far down a rabbit hole are you willing to go to essentially achieve the same result? But in hindsight, that's how I would do this again. And it's because of those 2 big elements: lack of information, and even when we state things, things don't play out the way we think they are. So I don't know if that's helpful for you, but I have thought about this question before, and it would answer a different question. It wouldn't answer the question we were trying to do here, which is how much do I know the literature and how much can I get the same result, however I define that, to do the exact same thing. I would reframe my question to say, how do I get somebody else to achieve the same result based on their interpretation of the data?
So yeah, Tim, I actually have a question. So what you did was actually you had a lot of information coming from the authors, right? Not from the paper only. It was all like public available and at the OSF, the protocols and everything. But what we're doing is more of we're taking the paper on its own, and the replications are actually blind, right, to the original results. So. And we are not currently planning to contact the original authors, right? So
well, we will, but we'll not tell. I mean,
so replicators won't have access to it. So what's your view on that? Do you believe, like in general, whenever you're trying to reproduce a finding, I mean, you can't really count on
on receiving external author information. Do you think as a goal, I mean, for science in general, I mean, we shouldn't rely on getting information from other, from original authors, or just sticking with a paper?
Yeah, no, that's a really good question. So, right, and so people have written about this. If they're blanking on the one guy's name, I should know it. Where they say that in order to do a good replication, you should always engage the original authors, right? And that's there's a lot to, like I said, there's a lot to be said for it, because there's so little information that engaging with the authors gives you access to a lot of information. However, because you can't always engage them, and because there's this element of why should I have to engage them? Because what happens if what if I can't get access to them for whatever reason? That we should be doing a better trade of having it be decentralized, because that is what science is decentralized. I don't think that, I think you can do something similar to what we're doing here, which is don't try to make it if you're concerned about the time or you're concerned about the engagement piece of it, don't make it so that it has to sit independent from your process of reviewing things. Intertwine it. So for example, we were doing informal review from authors before we wrote up our protocol, and then they were asked during eLife to also do formal review. And I think there was high overlap between our engagement and their engagement. If anything, they got more, because we would have some authors that didn't engage with us that engaged during peer review. So whether you do peer review formally through a journal, or whether you do it yourself, which I would recommend some form of peer review, even if it's just internal, that there is maybe a way to kind of walk the line and say, what if I attempt to engage the original author during that stage? What if I tell them my goal is to do a replication, knowing that there's going to be some modifications, but I'm trying to do something that is going to represent your experiment as faithfully as possible. Means I might not do the same reagents, right? However you want to make that flexible is up to you, I think. But that you offer the invitation within the bounds up front to say, basically, I know that this is more peer review, but it's of your own work, so it should hopefully be fast. And how about you guys conduct peer review while we're also conducting peer review informally before we start our project? And then you just take it in like you would anybody else. I think that's a way to kind of get both. And then you can track how many had authors peer review versus how many did not have original author peer review. But you don't make it the way we did it, which is we attempted to do it up front, right? We tried to do it as a step in our process. I don't know if that makes sense, but that'd be a recommendation, because I do think there's value in it. When you get it, it's really awesome. It's really, you get inside information that you'd like never would have picked up from the paper. But sometimes you could spend all that time and you get something and it turns out, geez, was it really worth all that time? Because maybe now I'm overly constrained in the question I'm asking, and I'm actually trying to ask a more generalizable question than I want to do the exact same thing all the time. So that's an architecture question that I'd throw back your way.
That one came up at the eLife round of review. So Tim was our reviewer for a feature paper at eLife. I think Rick Klein actually made the suggestion, well, maybe you could have like 2 labs trying to replicate without author information, and one of them did, which is right. I think it complicates the analysis a lot. So we're kind of like, maybe not. But it's an interesting, I mean, it's definitely an interesting question. I mean, our approach at the moment would be to contact the original authors, I mean, just to see what we can get out of it, and to know like how much is missing actually, but maybe not use that in the replication, but still you'd have a measure of what was lacking here, and just so that I should know. I mean, if you document our own protocols and you have the like more on the original protocol, you can tell like how much variation do you have in relation to the original one, which I think is a valid metric.
Yeah.
That said, we have the big problem. We're taking a 20-year window, right? So as you said, data decays with time. We had a 3-year window, and that was hard. I mean, to get data for experiments like 20 years out might be very, very complicated. So I'm not sure if we can count on the same engagement. So we're trying to depend as little as possible on that at the moment. We did incorporate. I mean, that was actually your suggestion at the start. But we did incorporate the round of peer review within the within our own network, which I think actually makes a lot of sense. And I think we've been working on those lines. Again, I think there's 2 different questions: whether you can replicate with information and without. I mean, both are valid. And I think we took the second approach, and for practicity as well. But I think it's I mean, both questions are interesting.
Yeah, no, completely agree with you. Yeah, I mean, I've been asked it before too, and I don't think there is a quote unquote right answer. I don't walk the line of saying do one or don't do the other one because they're interesting. I mean, to know, because it sits on two ends, right? The information, whether the information's actually, whether the change you're doing, right? Like if it turns out the author told you, oh shoot, you need to do X, and it turns out you guys weren't doing X, or maybe one of your labs did X. That's really good to know, because even though on an individual basis, it's not much you can learn from across all of your projects, you'll start to ask that question of, is it correlated, right? Is there a correlation between the missing? And I think that is a really important question, because I think we all want to believe we know exactly what's causing the effect, right? Because that's our research, but it doesn't mean that we do know, because the only way to know that is to actually have somebody else do it, to ask the question, do I get the same effect? And so you're always stuck in this, I think, middle zone of, well, do I need to have your information to do this or not? We also got asked a couple times, which is, well, shouldn't you do your experiments back in the original author's labs? And I was like, no, like, why should we be bound to the location of where it's done? Like, that doesn't make any sense. But you also get it, because there's an element of saying, well, when researchers can't figure something out, and if you're really invested in it, one thing you can do, and some people do, is to actually go visit the other lab and work it out side by side. But it doesn't mean that that should be a prerequisite for all studies in my mind, because otherwise we're never going to go anywhere, right? We can't all be around the entire world all the time trying to replicate each other's experiments. We need to be able to find a way to do it from our communication system. And if that's papers, we need to be able to work at least off of that as the backbone to everything. At least that's how I see it.
Yeah, and I think that also makes a case in terms of like having more studies on multi-center replication, which is one of our own tenets. I mean, even, yeah, I think we know very, very little about interlab variation. I mean, it's a huge issue, and there's not that much issue in that. So again, if we can contribute to that, I think it's also an interesting secondary goal of the initiative.
Yeah, I think that's brilliant. I think that's a way, and others have written on this too, and my thinking is the same way on this, which is, if you get back to how we do it again, which is you go down a pipeline of having somebody work it out. Once you figure it out, quote unquote, somebody figures it out, what you actually want to do then is say, can you do it across multiple labs? You should pull that clinical process from multi-site to single site down into the preclinical pipeline. It should come down, the concept's the same. You just don't invest on it up front, but you're right, you should be able to do more of it, so that way when you try to get more robust results, you're doing it in a more robust manner. And you do that by dispersing it across more groups. So I completely agree with you 100% on that. Awesome.
Anyone else out there in here? So yeah, I think we're pretty much done.
Awesome.
Thanks a lot thanks a lot for being with us. I think we have been learning a lot from you, not only today. I mean, it's like the third or fourth time to talk, but including the review. But I mean, it's just been great to count on you to learn on what worked, what didn't work, what I think it's we can definitely. I mean, when they say it's a large challenge, we can definitely relate. I mean, we're starting to do this, and we haven't even started the experiments yet. So I mean, we've been at this for a year, and just for planning. So I mean, I can fully understand the breadth of the effort that you have undertook, and the one you're going to take actually. I mean, he didn't really mention the next project, but it's probably much more of a very complicated beast. But we wish you luck, and I mean, if we can offer anything as well, I mean, we'll be happy to give it to give advice back, even though I'm not sure we.
Yeah, absolutely. I was saying, let's not lose touch there. And if it would be helpful if you guys keep doing this in the future as we get more clarity, I'd love to talk about the SCORE project with you guys too. I think there's constantly learning back and forth is the way to go on this.
Yeah, great. Well, so thanks, Juliana and people from Rede Pensadores for putting this up. And we'll be back. Just let me do the advertising here. We should be back on May 7th with our second webinar. So it's the first Tuesday of each month. Next speaker will be Malcolm MacLeod from Edinburgh, from the Camarades group, who's been working a lot with reproducibility in the preclinical field. He does a lot of meta research and attempts to do synthesis and meta-analysis in animal studies. So he'll definitely have a lot to say to us as well. So, and well, the May one will, so May 7th, 4 o'clock, same time, first Tuesday of each month, and we'll keep on going from there. So thanks, team. Thanks, Juliana. Thanks, everybody who's been here. It's great to share this information. We should do a lot more of this on debates. They're easy to do, and I think you'll learn a lot from them. So, yeah, hope to see you now all next month. Just as a final announcement, we are recruiting. Our final step of recruiting for the initiative finishes tomorrow. So we're just like getting some extra labs and specific expertise. But if you know anybody who wants to register still, you have like one more day until tomorrow. After that, we'll be definitely closing off, and we'll probably like be distributing experiments until the, I mean, certainly until the end of April to start working on the protocols. So we're moving, and thanks a lot, guys. See you next time.
Thank you, Olavo. Thank you, team. Thank you, everybody.
Bye.
Bye.